Okay, lecture 5 will discuss about the genome structures of different systems as well as the DNA replication process. Okay, first the structure of the chromosomes or genomes, if you will. So chromosome, by its definition, it's made up with DNA with associated proteins, and it's found from the living cells, of course. By its nature, it has a very compact structure which allows packing up a long DNA strand into a small space that is found within the cell, and especially within the nucleus. Okay, so to make long strand of DNA to be condensed so it can be stored into a small space, you need this compact structure. That's a chromosomal structure. The packed structure also protects the DNA from damages that could happen. Also, it maintains the DNA in a stable condition. Okay, and also the chromosome structure allows efficient passing to the daughter cells during the cell division process, and accessibility to DNA is regulated during the various stage of cellular events, okay, like replication or cell division, all those processes we'll talk about. And chromosome can be circular or linear, meaning, a lot of prokaryotic cells have circular DNA structure. We'll talk about it. But a lot of eukaryotic cells have linear DNA structure. Linear means there's one end and the other end. Okay. And prokaryotic cells have their chromosomes packed in a structure called nucleoid. That's a special term describing the chromosome-like structure in the prokaryotic cell. You don't call them chromosomes, instead you call them a nucleoid. And they carry, besides the chromosome, they may carry independent circular DNA plasmid, which are much smaller. Independent meaning, they are not part of the chromosomal DNA. The plasmid DNA is just kind of as separate entities, and they are replicated separate. Okay, so those are plasmid DNA. So we call them extra chromosomal DNA. And eukaryotic cells are mostly diploid. You probably remember the term diploid versus haploid. Okay, diploid means there are two copies of homologous chromosomes, right? And each chromosome carries a set of genes, so there are actually two copies of a gene found in diploid cells. And some eukaryotic cells are haploid, although most of them are diploid, but some examples are the sex cells, like sperm and egg cells. They only carry a single copy of a chromosome because they are united later on to double the number of chromosomes. Okay, these are all, I guess, the facts you already know of, hopefully. So I'll simplify structures of the prokaryotic cells versus eukaryotic cells, but they don't represent true size. And size, normally prokaryotic cells are maybe 100th or 1000 smaller than eukaryotic cells. Okay, so we are talking about nucleoid, that area, that structure where chromosomes are found. So they have kind of cytoplasm space where things are kind of all together. Okay, there's no membrane-bound organelle, of course, you're familiar with those. So this, I don't know, maybe looking like spaghetti noodles or together. Actually, if you stretch out, it's going to be a long big circle, one single circle. Okay, on top of that there may be these smaller balls. Those are plasmid DNA represented. Okay, so they exist kind of independently. And the eukaryotic cells, of course, you see maybe larger size, maybe 100 times larger than prokaryotic cells, or 1000 times larger, and they have membrane-bound organelles like nucleus, maybe mitochondria, or membranous network like endoplasmic reticulum, Golgi complexes, and things like that, right? And inside of nucleus you will find chromosomes, and they are linear, meaning there's one end and the other end, and if you stretch out, it's going to be a line of DNA strand. Okay, that's what you see. And then if you look at various chromosomes found in different living systems, from the prokaryotic cells to eukaryotic cells, all the way up to human. Okay, prokaryotic cells, like I said, they have small number of chromosomes. Mycoplasma, okay, have one chromosome and haploid, one copy, and they have circular DNA. Genome size only like 0.58 megabase pair. And E. coli, same, one chromosome, singular, circular, and then one copy of all genes, also circular, 4.6 megabase pair. Agrobacterium tumefaciens, they have 4 chromosomes instead of 1, but they are all haploid, one copy of genes, and they have 3 circular chromosome and 1 linear chromosome. It's unique. They have linear chromosome as well. And genome size about 5.67 megabase pair. And the last one is what? Rhizobium melanotoi. Yes, 3 chromosomes, 1 copy, haploid, circular, 6.7 megabase pair. And eukaryotic cells starting from fungi yeast, Saccharomyces cerevisiae, that's a baker's yeast, budding yeast. They have 16 chromosomes, a lot more than prokaryotic cells, and they may have 1 or 2 copies. The reason why it's 1 or 2 is yeast cells can exist as either haploid or diploid because they can fuse between 2 cells. When they fuse between 2 cells, they become diploid cells. Okay, so that's why it's 1 or 2 copies. And the structure of the chromosomes are linear instead of circular, much larger, 12.1 megabase pair. Another type of yeast is Saccharomyces pomb, fission yeast, 3 chromosome, same way 1 or 2 copies, meaning they can be either haploid or diploid, linear chromosome. And then we have Roundworm, C. elegans, 6 chromosome, 2 copies. It's definitely diploid from now on. So fungi, yeast cells are kind of unique. They can exist either haploid or diploid, but the rest of them are all diploid. Linear chromosome, much larger genome size. And plant, mustard family Arabidopsis thaliana, 5 chromosome, definitely diploid and linear chromosome. Drosophila, it's an insect cell, fruit fly, 4 chromosome, diploid, linear, 180 megabase pair. And then this is protozoan cell, again, of course eukaryotic cell, 5 chromosomes, 2 copies of chromosome, linear, huge genome size, 125 megabase pair. And so on, fish, as well as mouse, mammal, okay, another type, and then human. So we have 22 number of chromosomes and 1 sex chromosome, either X or Y. And then we have diploids, so total chromosome is actually 44 plus X, double X or XY, so total 46 chromosomes, right? Linear, 3200, I guess, megabase pair, much larger. Okay, so understand difference among the chromosomal structures of different living systems. So if you look at those genomes, okay, the size are different as we just saw, but the complexity of the genome is also unique. Okay, if you have a small genome, like maybe mycoplasma or viruses or E. coli prokaryotic cells, although their genome size is small, they have very dense or compact genome, meaning within a small size genome they have a lot of genes together, so there is no waste of their genome. Mostly genome is made out of mostly coding regions. Sometimes some genes are even overlapping, so it's very condensed genome. That's what you can see, complex genome. And viruses have highest gene density, meaning within a small size there are a lot more genes found. So often you can find overlapping genes, and within a same part, multiple genes may be encoded. That's what overlapping genes is. So here, E. coli versus yeast. So if you look at maybe small area, how many genes can you see? So RNA polymerase coding gene is found here in the E. coli cells, and fungi here, and what you see is a fruit fly. You may have now the blue parts are introns, so there are non-coding regions kind of intervening the coding region, exons, right? But fungi and E. coli, they do not have such things. These are all coding regions. Human, of course, have a lot more non-coding regions. So within a given size of genome, how many genes can you find? That's the gene density you are talking about. Within the same size, there are only 2 functional genes here within human genome. But here, how many genes? In the fruit fly, 9 genes can be found. And fungi, 31 genes. And E. coli, 57 genes. That's what you mean by a lot more dense genome. Okay? So that makes sense because within a small genome, if you want to have all essential genes, you have to use your genome very efficiently. Okay? And eukaryotic genomes have increased intergenic sequences. Intergenic sequences meaning, between the coding regions there are non-coding regions. We call those intergenic sequences or introns, non-coding regions. Okay, so prokaryotic genes, of course, they would not have a lot of intergenic sequences because they don't have enough space to waste. Okay, so prokaryotic cells almost like no repetitive sequences and no introns found because there's no space that can be wasted in the prokaryotic cells. But on another side, eukaryotic cells, fungi, all the way up to plants and vertebrates, including human, gene density is much lower, and then average number of introns found per single gene, there are a lot more number of introns found in within a single gene. So, and then not only that, there are a lot more repetitive sequences. Repetitive sequences normally not really functional gene coding sequence. They are just found there for, I don't know, the reason that we do not understand. They may have some important functions, but we don't know what they do. Okay, but there are a lot of those repetitive sequences in the human genome or higher advanced living systems. Okay, so look at it. Human, where is it? 46 repetitive sequences are found. That's a lot. Okay, so that's the trend you can understand. And then the majority of human intergenic sequences are made of repetitive sequences. Okay, which includes microsatellite DNA. Very short tandem repeats. The size of the repeats are about 13 base pair long, and you have repeats of a lot of those. Okay, and there are genome-wide repeats that are larger repeats, larger than maybe 100 base pair or 1 kilo base pair or larger. Those are also found, and many of them are transposable elements. Okay, so-called jumping genes, and they may move from one location to another. There are enzymes that can help the transposition. That's the process called, how these transposable elements can move from one location of the genome to another. Okay, so human genome, so gene coding region is only this much, but intergenic sequence is about 2,000 megabase pair. Genes and gene-related sequences are only 1,200 megabase pair. And then other intergenic regions, 600 mega, and genome-wide, repeats. So these are all seemingly kind of non-functional parts of the genome. And then genes only take up 48 megabase pair, okay, out of the entire genome size. Very small, only almost like 1/10. And related sequence about that big. Introns, genomes, that's including introns within that size, okay, because they are found inside of the gene coding regions. Gene fragments, pseudogenes. Pseudogenes are kind of gene-looking genes. They have like a start and stop codons, but they are not expressing any RNA or DNA. Those are pseudogenes, okay? So I'm going to stop right here.